microfluidics in one lecture. So I'm just going to try to give some basic principles of what the advantages are, why you might want to use these devices in your own studies, what some advantages are against other approaches, et cetera, with the idea of getting some general principles across to you, along with how do you make these things. One thing my group does quite heavily is collaborate by making devices for other people's approaches. So if there's anything in here you find interesting or advantageous for your own research, please don't hesitate to reach out to me. And we can talk about making devices, particularly if you're from a lab where microfabrication may not be a core technology. And I'll also go over some of the challenges with designing and using these devices for mechanobiology applications. So I'm going to start by giving you guys a little bit of a hangover to a time we all thought we left behind. But I imagine all of you have become, whether willingly or not, quite familiar with these devices, these rapid tests for COVID. So I think some of my colleagues might yell at me and get real worked up whether or not these are microfluidic devices. But as far as I'm concerned, these are some of the most pervasive microfluidic devices. And sure, maybe the length scale is more millimeters, but the operating principles are still shared with microfluidic devices more broadly. So I think they serve as a good teaching tool. So I know it's up here on the slide, but somebody in their own words tell me what you're looking for when you use one of these devices. Like physically, what are you looking at this piece of equipment to tell you? What was that? I heard a mumble.
So yes or no to an antibody being present or not?
Yeah, and so how do you tell if the antibody is present or not?
Looking at the two lines.
Right, looking at the two lines. So the endpoint, especially this line here, the test line, right? The endpoint of this assay is a specific chemical reaction that occurs with high spatiotemporal resolution, right? So, and this is really key to why these specific devices are used for these at-home rapid tests, because they allow for this high spatiotemporal resolution that lets somebody at home easily determine whether or not they have the antibodies against the SARS-CoV-2 antigen. So this kind of is the central theme of the use of microfluidics, that they provide this high spatiotemporal resolution over chemical reactions, but also, as we will discuss later, the application of mechanical forces. And this coupled with this high spatiotemporal resolution is the highly repeatable result. I mean, we can debate about what highly repeatable means compared to other assays, but generally speaking, once a microfluidic assay is standardized, one of the advantages are they tend to be highly repeatable. So when you're using one of these COVID rapid tests, for example, what's the first thing you do to prepare a sample to be tested? We can use this kind of device to talk through some of the benefits of why a microfluidic device was chosen to do or developed to do these rapid testing. So what's the first thing you do when you go to take a rapid test?
Wash your hands.
Yeah, okay. Actually, that's a good one. Yeah, so wash your hands. I'll come back to that in a second, but then there's something else you do that I don't know, depending on if you're doing it to yourself or someone else to you, it makes you cringe or not.
Get a sample.
Collect the sample.
Yeah, collect the sample. You stick a rod up your nose and you move it around. Right? So if we think about the end product design for what's going to test whether or not there's this SARS-CoV-2 antibodies in the patient, we need this sample collection, but then if we take this sample and we put it in the device, we need to make sure that the actual physical components of the device are not messing with our assay. So the washing of the hands is actually a key part of this. We need this device itself to be sterile. The fact that you have to wash your hands demonstrates that it is sterile, because you don't want bacteria and viruses that are on the surface of your skin contaminating the assay. So what we need from a device or a system to test from SARS-CoV-2 at the most basic level, it's obvious, is this biocompatibility, so that these materials are not cytotoxic. They don't kill. In this case, in the SARS-CoV-2 assay, we're not really caring about mammalian cell survival. But if you're using these devices for mammalian cell mechanobiology, then you may want to make sure that the devices do not kill your cells that you're seeking to assay. And you need to be able to sterilize them so bacteria, fungus, and all the other good stuff in the environment don't contaminate your assay. And that turns out to not be a trivial component to the devices in their actual use, as we discuss later on. The second thing you'll do once you extract this sample is you swirl it around in actually a pretty low volume of extraction buffer, right? This is, something on the order of less than a milliliter. And so another key requirement is we need to have small working volumes. We don't want to have to extract a piece of tissue. We don't want to have to take a blood draw from a patient. This is part of the purpose of this device, is to make it easy and accessible at home. And so we want to work with small samples. And if we're working with small samples, then our assay has to be performed in small working volumes. Again, this is maybe obvious because the subject of today's talk is microfluidics, which implies small working volumes, but still for an assay like this, it would be necessary. The second thing that's really important, and maybe a little more challenging to understand once you get under the hood, is that we need very precise control over pressure and flow. So the reason we need it for an assay like this is we are looking for the presence of antibodies at a very specific point in space, at a working time that is reasonable for an end user, something like 15 to 30 minutes. But the rate of the reaction that's leading to the red line appearing has to occur at a rate compatible with the flow of the sample going across my test region. If I'm flowing my sample across too quickly, I may get a false negative. If I'm flowing my sample across too slowly, I might get a false positive. So inherent in the design of these devices is some fluidic principles that allow really precise control over pressure and flow, and that is a requirement to many microfluidic assays. Along with that, we need these devices allow for a relatively rapid reaction time. So because the small working volumes and because we have precise control over pressure and flow, we can allow this readout to occur on the order of 15 to 30 minutes. We know that often this 30 minutes is longer than is necessary. So these devices afford a rapid reaction time. And then in terms of reading the results, the devices need to be optically transparent so we can see them and afford the high spatiotemporal resolution, which, as I said on the slide before, is kind of the organizing principle of this whole talk. So if we take all of these as sort of requirements for the functioning of this device, we can see why this kind of microfluidic approach might be taken, because it satisfies these 5 key components, but also we can use these principles to discuss why microfluidics might be helpful in mechanobiology. One quick note is that although my lab's expertise and my research for most of my career has been on microfluidics for mechanobiology purposes, there are many, many applications of microfluidics. And in fact, you almost will certainly use a microfluidic device in one form or another in your own research, in your PhD. These are just 2 examples. One is single-cell RNA-seq, uses droplets that include, the specific approach varies from commercial system to commercial system, but basically you form droplets with some kind of barcode in them along with your cell, and then you can do sequencing out of them. The key enabling technology for single-cell RNA-seq was the microfluidic devices. Fluorescence activated cell sorting, something that's used to separate cells based on fluorescence signal. This also was an early kind of microfluidic device for biological purposes. But even if you don't work with cells or do use either of these approaches, you will probably print something at some point, and probably the most pervasive microfluidic device in commercial availability is inkjet printer heads, which use actuators and phase change of inks in order to print with really high speed and resolution. Okay, so why would these specific platforms, these microfluidics, be helpful in mechanobiology? Well, to provide an example, I'll use a case study of metastasis of solid tumors. Okay, so I imagine most of you know, probably several, perhaps most of you are working on cancer in one context or another, given how important mechanobiology is in the progression of cancer. So this is probably a review for you, but for solid tumors, particularly those of epithelial origin, so carcinomas, metastases, or the formation of tumors distant from the primary tumor, are the leading cause of death in most solid tumors, but particularly in breast cancer. This slide on the bottom shows a patient before and after chemotherapy, and you can see a reduction in the mass of the tumor in the breast, but this patient, despite the shrinking of the primary tumor, this patient died of a metastasis to the brain. So there's a really compelling reason and interesting science on understanding how cells go from a primary tumor and escape to colonize distant tissues. There's a lot of mechanics involved in this process, but one of the most fundamental requirements is that cells, which normally in a healthy epithelial tissue, normally form this kind of blocky structure, this brick-like structure to maintain barriers. That's what epithelial cells do. They acquire the ability to migrate, to exit the primary tumor, cross some matrix that's at the primary tumor, enter into either the blood or the lymphatics, and survive the blood and lymphatics and escape to a different part of the body, get out, proliferate, form a second tumor. So from a mechanics standpoint, there is a required transition that happens in these cells. These blocky, static epithelial cells look like bricks, but in order for them to get into the vasculature, travel around, and then get out of the vasculature, colonize tissues, migrate, they have to squeeze themselves through various different interfaces. There's a guy at Cornell, Jan Lammerding, who's done really cool work on just studying this squeezing effect. And there's many other, Dennis Disher, of course, at Penn. Lots of cool people have worked on this squeezing problem. But we can maybe abstract it to discuss why microfluidics would be helpful by just thinking about this mechanics transition that must happen. That metastatic tumor cells have to squeeze through things. So perhaps a reasonable hypothesis might be that they're more compliant than the healthy cells, which form this brick-like structure and maintain a barrier in a healthy tissue. So if this was a hypothesis, and let's say you wanted to understand what genes are differentially regulated in these migrating tumor cells so that you could develop new drugs that target those genes, you might want to design an assay that would allow you to test to see if the metastatic tumor cells are indeed more compliant, more squeezable than the healthy epithelial cells. So if that was the case, if that was your hypothesis, that these metastatic cells were more squeezable than the healthy cells, what would you need? What would be a reasonable way to test this hypothesis? How would you test the squeezability of cells? Somebody give me a suggestion.
Could you let the cells flow through like chambers that are like decreasing in size or something?
Yeah, that's exact. You jumped right to the answer I was hoping you would. So you take a cell and then you either let it migrate on its own, or if you're just looking at the cell mechanical properties, perhaps you would want to apply a kind of pressure gradient on one side. So hold that side at a high pressure, hold this side at a lower pressure, and then design a channel that starts to decrease in size. I'm terrible at drawing, as you can see, and force this cell through. What we would expect is that if we measured displacement on the x-axis versus time for a given pressure gradient, we might expect that a healthy cell will look something like this. It might get in a little bit, it might make it into this first chamber and then get stuck. Whereas a really squishy cell, a cell that has the ability to migrate through pores in the matrix, but also through barriers in the vasculature or the lymphatics, it might make it further down the device, and we might see trend going further up. So this would allow us a device like this, if we could make these structures to constrain a cell and also to apply pressure, it would allow us to quantitatively test this hypothesis to see if metastatic cells are more compliant, assuming we can isolate the metastatic cells separately from the healthy cells. But then it would also perhaps allow us to sort the cells. We could run this assay for a little while. We could extract cells from this chamber versus that chamber, and we could run them out for sequencing and see what genes are differentially expressed in the two cells. We could also do genome sequencing to look for mutations that might subject certain papers to metastasis. Okay, so what will we need in order to make an assay like this? Well, the first thing we would need is biocompatibility. We would need the ability to extract a sample from a cell, or from a patient, to force them through our chamber of decreasing sizes. We would want small working volumes because metastatic tumor cells are rare, and patients who have metastatic disease are already suffering enough. We don't want to have to take out a bunch of tissue in order to run a test like this. We obviously need high precision over pressure and flow. After all, the assay that was proposed on the previous slide involves looking at cell deformation under an applied pressure. So that's very important. We want a relatively rapid reaction time. Cells are not too happy when you take them outside of their native microenvironment. So we don't want to introduce biases associated with cell culture or culture within a non-native environment. So we want to run our assay pretty fast. And if we're measuring the deformation of cells in response to an applied pressure, then obviously we want optical transparency to measure the deformations, and high spatiotemporal resolution is a given. We're talking about assaying individual cells. So remember that these were all of the motivations to use a microfluidic device for a rapid test, and they also suggest that a microfluidic device might be right for this specific mechanobiology application of measuring the mechanical properties of cells. One of the challenges, though, with making such an assay is the characteristic length scales of these cells. So this is an example from my own work when I was a PhD student, looking at the migration of these metastatic cancer cells. So this is a line isolated from a patient with metastatic disease, and it's a little hard to see because I wasn't very good at imaging. I also didn't have great microscopes at the time, but what you see here is this is where the cell starts. This is where it ends after about 10 hours migrating through a 3D collagen hydrogel. But what I want to draw your attention to is that this length scale, the entire width, like the entire extended length of the cell here, is about 10 microns. So if we want to make a platform that allows us to squeeze a cell, then we know that we need this chamber, which might catch the cell but not squeeze it, has to be on the order of 10 microns. That's very small. So creating a device that then allows us to squeeze further, let's say make this something on the order of 5 microns or maybe even less, this is quite challenging to do, to pattern physical structures that are on this length scale. And so there have been a number of techniques developed to create structures on this length scale. This slide, this is a review paper from my lab that was discussing different ways for making microfluidic devices. And these days, 3D printing is quite accessible. Most incoming graduate students know how to 3D print and even mill materials. However, if we wanted to get things on the order of 10 microns, smaller than 50 microns, then we're limited to the use of techniques called photolithography and a second step called soft lithography. These, every few years, you'll read review papers to say that these techniques are falling out of fashion, or the 3D printing is getting so good we don't need to use photolithography, but it still remains, photolithography and soft lithography still remain the best method for fabricating microfluidic devices with sub 50 micron length scales and with high repeatability. So I'm going to go over how these things are made and eventually go back to showing an example of how we could squeeze a tumor cell. So photolithography, the process by which we pattern materials in order to make structures that are smaller than 50 microns in diameter. The name, I think I have actually I'll move ahead and come back to that slide. This process is called photolithography. It's been around for a long time. The name is somewhat self-explanatory, but it comes from the idea of using light to write on stone or a really stiff material. It was first discovered or developed in the 1820s, and lots of people made a lot of money very quickly off of photolithography in order to generate the first photographs, but also printing presses at the time. The first photolithographic material was this bitumen of Judea, which was discovered in the 1800s. It's a naturally occurring asphalt-like material that becomes insoluble when exposed to light. So what you could do with this material and what they did to develop the first photographs is you could expose bitumen of Judea to different intensities of light. And the regions that saw the highest intensity became insoluble in certain solvents. And thus, if you expose to varying intensity and then dipped in the solvent, you would get a extrusion that was as deep as the thickness of the bitumen of Judea, which reflected the light intensity that was exposed on it. The reason that this approach of using light to pattern a material is quite helpful for biological applications is that the visible light wavelengths are similar in length scales to a virus, or smaller than most mammalian cell structures. So if we can pattern light, then we can achieve very high resolutions, things much smaller than cells or other biological materials. So in theory, patterning light provides us with resolution necessary to poke and probe mammalian cells or other biological materials. So the way we use photolithography to make microfluidic devices in the most standard approach, there are thousands of variations to this approach that have different pros or cons depending on your specific application. But the most general and also most widely used method of making microfluidic devices with photolithography is a two-step process. The first is to use actual photolithography to pattern a silicon wafer. We use silicon for two reasons. One is that silicon is the most used material for photolithography because it also forms, this photolithographic process forms the basis for how microprocessors and integrated chips are made, the things that allow our phones to be so small and our computers to be so thin. So there is a multi-trillion-dollar industry in figuring out how to pattern silicon, taking advantage of its semiconductor properties, and we can leverage all of the research and technological developments done there in the microchip world to make better microfluidic devices. But broadly, this process of photolithography provides the high spatial resolution and low working volumes that were the requirements for this assay that were laid out on the previous slide. And the first steps involve coating with a photoresist. So this would be like that bitumen of Judea material that was discovered in the 1800s. We now use these polymers such as the SU8 class of polymers. These are materials that are specifically designed so that if they are exposed to light, they become either insoluble or more soluble in a solvent, depending on your workflow. The typical photoresists we use are what are called negative photoresists. And these become insoluble when exposed to light. So what that allows us to do is make a photomask like what is shown here. And a photomask simply allows light to go through at some spots, blocks light from going at other spots. So we put the photomask on top of a photoresist coated wafer and we expose to high intensity light. and regions that get hit with light get cross-linked and become insoluble. So then we can wash them out. In this case, it's a little confusing, but the green spots are blocking the light from being present. So they're not, they don't form structures after development. And then we can, the problem though with this approach is that one, silicon is really stiff, and two, photoresist is not very biocompatible unless it's treated. So what we often do in the microfluidics realm is we just use this patterned silicon wafer as a negative mold for the eventual microfluidic device, which is created by pouring on PDMS or some other polymer and letting it cure on top of our features, peeling it off, and we end up with an inverse of what was patterned on the original wafer. The reason we use PDMS, polydimethylsiloxane, is that it gives us the biocompatibility and optical transparency, which were requirements of our original assays. After this replica molding process, we can punch out holes and connect devices and stuff to allow pressure and flow control. Okay. Before going on, does anybody have any questions or thoughts? No. All good. Okay. I can't pick on you without being able to see you. So anyway, let's go back to the original problem here, which was to create a device that allows us to squeeze cells on the order of 10 microns. And I'm going to show you now that how we've designed such a device. I should state that everything I'm about to show you is done in my lab, but was copying, like directly copying work by Jan Lammerding at Cornell. So he deserves all the credit, but you'll see screenshots and stuff from our own lab. So Anya, a technician in my lab, wanted to squish cells under a similar hypothesis to what was laid out for the cancer cells, but actually we're using a slightly different cell type that I'll explain in a second. And so to make this device to squish the cells, we need to first design a photomask, the photomask being this component, which is going to block or allow light onto an SU8 coated wafer. And we can do that using technical drawing software. We use AutoCAD, but you can use Illustrator if you like Illustrator. You can use, there's various other microfluidic specific software you can get, but I guess I'm old, so I like AutoCAD, which I think is coming out of favor these days because it's expensive unless you're in academia, but it's pretty easy to use. So this shows a line drawing of the masks that we're making to fabricate these microfluidic devices. And what you'll note is that at this length scale, which is shown on the left side here in the screenshot of AutoCAD, we can't even see the region where the action is happening, where the squeezing is going to happen. That's all down in here. What you see at this length scale are these kind of weird trapezoids with a bunch of circles inside of them. These are our pads to connect pressure. So the way this assay is going to work to squeeze the cells is we're going to have two pressure inlets. Pressure inlet 1 here, pressure inlet 2 here, and then a pressure outlet here. If I zoom in on this red region where the action happens, you see some little yellow lines. That's what's shown blown up over here on this side of the slide. You can see it right in there. If I zoom in a lot, I have a bunch of these pockets. And these pockets are about 20 microns to catch the cell, and then 5 microns to squish the cell. There's a little channel sticking out. The reason that these pressure inlets have these structures with all of these little circles inside is simply because from pressure inlet 1, we are going to flow in cells. And the cells that we're using in this assay, again, they're not metastatic cancer cells, but they will be when we run those tests eventually. But right now, these cells like to stick to each other. So what we have here is like a little pachinko game so that individual cells have to bounce through before getting into this channel and then getting squished in this direction in that region. And these little bouncing around actions help to separate individual cells and make sure that we're getting one cell at a time filling these little pockets. This is also why we need 3 pressures. We need a pressure to introduce the cell solution to get them to flow this way to fill our chamber. And then we need a pressure gradient going this way to squish them, to push them through these constricted channels here. So what we have done on this slide is we've created the masks that achieve the geometries necessary to test the mechanical properties of these cells towards that differential squeezing hypothesis. Then with those masks, we can go to the clean room. This is Harry. He is not making the squishing device, but Anya, the person working on it, was too self-conscious to send a selfie. So we stuck Harry in there instead. But part of the reason I show Harry is because he is an undergraduate in our lab who learned these techniques in a semester. And I just want to use that as an example, not to say that Harry was not an extremely talented person. He's now doing his PhD at Caltech, but to show that these procedures to make microfluidic devices might be intimidating at first glance, but they're quite accessible, particularly if your university has a clean room facility, or if not, if you have a collaborator or other institution that can provide some training. These techniques can be learned in a matter of weeks. But what Anya, who is not Harry in this picture, did was she took her masks and she went down to the clean room and she spun on that SU8 negative photoresist. And when I should say explain that term spun on. So the way that we actually, so one of the important parts in the functioning of this device is that it certainly has to constrain the cell in this direction, in what we might call the XY direction. But we also need to make sure that coming out of the page, that this device is not so thick that it's not squishing the cells sufficiently. So actually, if we look in the cross-sectional dimension, the channel not only constricts down in the XY direction, the XY plane, like I showed before, but if I look in, let's say, the XZ plane, then I also have a constriction that makes sure that the cells are being forced through an area that's 5 microns in both key directions. So this region, the squishing kind of channel, is actually a bit smaller or thinner than the incoming channels that catch the cells. And the way we control the thickness of that photoresist to make structures in different heights is we take a silicon wafer, and now if this is the side of the silicon wafer, we stick it on a machine called a spin coater that rotates at a known angular velocity, omega. Now, if I plop down a bunch of photoresist on top of my silicon wafer on a spin coater, then it turns out if I know the viscosity of the photoresist, then the angular velocity that I'm spinning the wafer will determine the resulting thickness of the photoresist for a given viscosity. So when you buy photoresist, they have these big tables or charts that show you spin your wafer at this rate and you end up with photoresist at that rate. If any of you have been in the clean room, you know those tables are meaningless. You have to repeat them yourself, but either way, there is some relationship between the spin rate and the resulting thickness. So this is why these SU8 materials are really popular in microfluidics for mechanobiology, because they're what allow us to get thicknesses on the order of 10 microns or that of a single cell. So when I say Harry or Anya spun on photoresist, that's what I mean. They loaded it on one of these spin coaters and they rotated it at a given velocity. And they exposed their coated silicon wafers through those masks on the previous slide that was shown here. And they ended up, after developing, with a structure that looks like what's shown here. This is kind of the final device on silicon zoomed in on the area of action. Then we used soft lithography with PDMS to create the devices here on the right. You can see that they're very small and that the 5 micron region you cannot see by eye without magnification. But if we do that magnification just on a wide field microscope, you see we have really nice features. We have great resolutions. If you measure these dimensions, each one of these channels is indeed 5 microns, as you can see below. So to further characterize the length scales, we load this up with a fluorescent solution, use confocal microscopy to look at the dimensions. This shows why I'm maybe better as a PI than actually as a technician, because this was the device that I tried to make, and you can see that the squishing channel is not in the center, but Anya's device, which is up here, it's just beautifully centered itself as a manual error on my part. But you can see that the dimensions match what we wanted them to be from the original mask design. So the process of going from the photomask itself down to the final device is not that challenging. It requires a little bit of practice, but then that means that the challenge in the microfluidic device design is in generating the appropriate photomasks. But just to show a little bit of data of what these things look like is that these are actually HUVECs in this case. These are human umbilical vein endothelial cells. So they're not the cancer cells that motivated this process, or this discussion. But you can see there's a couple of channels where there's some defects and cells fly through really fast. I don't know how well this movie is playing on your end, but for the most part, these cells kind of get stuck at one specific location. They're all sort of in this region over here. Whereas these are mutant cells, these are expressing a mutation in the PI3K pathway that would be associated with cancer. And when we throw them through this device, you can start to see that one, they look a lot bigger at baseline. That's what my lab is studying right now. But also you can see that they extend much further into these constricted channels. I actually think the magnification is slightly different on these images too, because it's the same device. But either way, if we plot, like I had originally suggested and had been suggested by one of you, the protrusion length, or the length into the channel as a function of time for a given pressure gradient, where we know that this is P1 and P naught, then I can see that my mutant cells indeed squeeze through faster than my healthy cells. So this demonstrates that indeed that all of the physical requirements that were stated on the previous slide allowed us to measure the mechanical properties of these cells, and sort of illustrates how microfluidics might be useful broadly in mechanics studies that involve cells or collections of cells. But there is one problem with applying these approaches if you think about modeling mechanics of the native microenvironment. So here we wanted a device that was essentially infinitely stiff compared to cells, because we wanted to constrain the cells themselves. We wanted to push them through a channel, and that channel not to deform in response to the applied pressure that's inherent in our assumptions underlying the analysis to measure the compliance of the cells. But if we're going to actually simulate mechanobiological processes, like if we want to see cell-generated forces, for example, or we want to see tissue morphogenesis, then these stiffnesses are way too high. There's nothing in the body that, well, other than bone, that's that stiff, and very few cells that you all will study as part of your PhD experience stiffnesses this high. So one of the more recent, but is quite pervasive now, sort of subfields in microfluidics for mechanobiology is what I broadly call porous media microfluidics. And these are microfluidic devices that also involve the use of a three-dimensional hydrogel. So I know Jason Burdick is speaking next about hydrogels and hydrogel mechanics. And so it turns out that with microfluidics, we can leverage the sort of pressure and flow resolution, the rapid reaction time, the low working volumes. And if we can integrate a three-dimensional hydrogel, then we end up with something that also has all the benefits of hydrogels, which include the things that you'll hear about in the next lecture, but things like physiologic compliance, the three-dimensional structure, cross-linking, reaction times, et cetera. So just a quick note on how we actually introduce hydrogels into microfluidic devices, because this is a good transition between both the benefits of microfluidic devices, but also one of the key challenges, and that is surface tension. So surface tension is a force that is present at solid liquid interfaces, solid liquid air interfaces. It's something that we experience day to day, but is quite interesting and complex in the governing physics. You all know this if you ever belly flopped into a pool. The reason it hurts is because surface tension of the water needs to be broken in order for you to enter the body of water itself. So there is some energy stored in the surface of liquids, and that deforming the surface of the liquid can provide an upward force that counteracts things like gravity or pressure. In this case, is keeping this bug, a water strider, afloat. So I'm not going to go into the details of surface tension for the time being. Hopefully, most of you are generally familiar with the concept, but basically, surface tension is also what gives rise to air bubbles in your beer carbonated drink. And you can figure out the magnitude of surface tension by doing a simple force balance by calculating the pressure inside of a bubble, for example, as compared to outside. And the difference in these pressures is due to the fact that surface tension is acting along the air-liquid interface in parallel to the air-liquid surface itself. And the important point here is that the pressure differential that can be maintained for a given magnitude of surface tension, which is dependent on the air-liquid interface itself, is inversely proportional to the radius. So in other words, very, very small radii of bubbles or other air-liquid interfaces lead to very high differential pressures from inside to out. So that, in a very clever way, has been leveraged originally by Newly John and Roger Cam to constrain hydrogels into specific regions within a microfluidic device. So the way you can introduce a hydrogel into a microfluidic device is you go through that process of photolithography and soft lithography, like I just went over in the previous slide. But you include a bunch of these little trapezoid channels, these sawtooth-like structures that are in the center of the center channel within this device. This device has 3 parallel channels, which can be seen in various views here. But dark red, kind of gray, and light red here. These are the 3 parallel channels. And then it's the sawtooth structure that separates them. The spacing between those saw teeth is narrow enough that if I inject a liquid hydrogel, there is an air-liquid solid interface and surface tension between the posts. So that keeps the hydrogel, or resists whatever pressure I'm using to fill. The injection pressure of my pipette, if I'm injecting a hydrogel by hand, is resisted by surface tension at this interface, and that keeps the hydrogel present into just the region here that is flanked on either side by these sawtooth-like structures. So this is a phase contrast image. So what I end up with is the hydrogel just in this region, and then I have flanking side regions that don't have hydrogels in them, and I can use that to set up molecular gradients or pressure gradients to look at the effects of different molecules, on cellular processes within the hydrogel, etc. So
I have a question for the
Sure, yeah.
microfluidic.
Yeah.
So
yes,
you mentioned that the main purpose of this design is to, I guess, prevent maybe fluid flow into a certain channel. Why is
Yes.
it, why are the pillars specifically trapezoidal? Like, why not rectangular or circular if really the distance between the pillars is like sort of
that's an awesome question. And I should say that this is a little bit older work. It's kind of like a good teaching example, but our lab has gone away from the trapezoids. So if this is something that is relevant to your research, then please reach out and I can explain why we have.
Yeah.
But the reason trapezoids are used here is because actually of what's shown in this graph here. If you have rectangular structures and you fill with your hydrogel solution, then what you'll end up with is, like if the hydrogel's in here, you'll end up with this bow-like, this bubble-like structure that sticks out into the flanking channel. And these specific devices, which are depicted here, are made so that you eventually seed this channel with cells, and that little bulge that occurs between the square channels kind of makes seeding annoying. I was in Roger's lab at the time they were working on this, and I wasn't working on this project, but lots of people go through all the complexities of making the device and then wouldn't be able to seed very well because of these bubbles. So this device, the trapezoid itself, is designed so that the angle of each trapezoid matches the contact angle of water on PDMS that surface treated as theirs is. And that allows for this flat interface, which if I erase these lines, you can it's hard to see on this image here, but there is a flat hydrogel interface that can then be seeded with cells. Does that answer your question?
Yes, thank you. We actually, my lab received microfluidic molds from the Roger Cam lab. And so
yeah.
we were trying to like reverse engineer or reverse explain like the design. But like the publications available didn't really dive deep into the rationale. So thank you.
Sure. Yeah, no, actually this one is overlooked a lot. It's in +1. I think that's part of the reason why. But this guy, Waleed, the first author, was really the one who brought these devices to the CAM lab. And he was a hardcore mechanical engineer and hated cell culture, and between us wasn't very good at it either. So this paper, like, because it doesn't have really any biological results, gets overlooked in Roger's publications, but I would look to that for motivation. If you have issues, so one of the problems with these approaches is if you are going to put cells out here, then the cells really respond to the stiffness gradients of the post versus the hydrogel. Like you'll see cells crawl along the posts before they get into the hydrogel. And I was going to show this as an example. I don't know, maybe because I came from Roger's lab, I feel a little bit more comfortable showing some of the blemishes, but anyway. But so I was going to show this as an example, getting back to the squishiness of cells, but they use these devices to make little vascular networks. That's probably the most common application of these devices. And here you're looking at a vascular network that's between two posts, like this entrance area. This is a very fortuitous image. If you try to do this yourself, you'll find a lot of cells like to line these pillars. And also that when you get beautiful vascular structures like this, it's most likely that one surface of these cells is in contact with the bottom glass. So anyway, if you're interested in using these for your own work and having trouble with the posts, feel free to reach out, because again, we kind of use these posts sometimes because they work easily, but then we don't like to use them. We have to seed cells along that parent channel because of the stiffness gradients of PDMS.
Question?
There's another question?
Yes.
Yeah,
the question,
go for it.
when you're using the hydrogel and the microfluidic system together, do you have a control about the pore size of the constriction pores?
I didn't. I heard you. I got. If you're using a hydrogel in the microfluidic device, and then I missed the second part.
Do you have a way to control the pore size?
Oh, the pore size. Yeah. So I see Dr. Burdick is on the call. I'll let him answer the complex questions about hydrogels. But yeah, you do. If the hydrogel you're using affords some control over pore size. But I should say, so jumping to the challenges with microfluidics, I don't know what slide best indicates this, but we can just hang out here. One of the problems with introducing hydrogels into microfluidics is many synthetic hydrogels in particular swell after cross-linking. So PEG, for example, PEG's great, control mechanics, you can present all sorts of different ligands, you can make it MMP labile, but it will swell after cross-linking. And a problem with these introducing that into a microfluidic device is if you inject that into the central region of a device like this and it's constrained on all sides and then it goes to swell, those stresses are significant. If you try to do that with a PEG gel in these devices, we've done it, you'll crack the glass that forms the bottom of the microfluidic device. And so that gets at your pore size question. So as long as you have a hydrogel that does not significantly swell, or if you have design features to account for the swelling, then indeed you can control pore size as you would with another hydrogel, for example, through cross-linking or other mechanisms.
Thank you.
Does that answer your question?
Yes, thank you.
Okay, cool. All right, so I'm going to skip over some stuff here just to show that this was just an example to show these are, this is like a tube of endothelial cells. That's kind of a core assay that our lab uses. I will go back one slide actually to show the basis for this. So one of the kind of devices our lab uses these days is we make these tubes of endothelial cells that we can then perfuse with pressure and flow. So by integrating the hydrogel with the microfluidic system, we can sort of model a blood vessel in which we can actually flow stuff through because we have control over pressure at the inlet and at the outlet. And these vessels are fully embedded in a collagen hydrogel. This is showing a kind of 3D reconstruction where green is collagen. So we can leverage the physiologic mechanical properties of the hydrogel, or the collagen in this case, and the pressure and flow to get a kind of perfusible microvessel. And this example, this just shows what one of these microvessels looks like when it's static, under static culture versus flow. Not a great image because everything drifts, but what you can see is that as we apply flow, in this case from left to right, the diameter of this vessel decreases. The reason I use that as an example for this kind of porous media microfluidic devices is because obviously we need a way to apply pressure to get flow to go from left to right. That's afforded to us by the microfluidic housing platform. But then this hydrogel is sufficiently compliant that the cells themselves can generate tractions necessary to close up the vessel to decrease the diameter, which is a very important mechanism of barrier function regulation in these devices. So with the integration of these two approaches, the hydrogel and the microfluidics, we kind of get all of the benefits of microfluidics we discussed before, but then also the features of hydrogels, which I'm sure Dr. Burdick will talk about on the next slide. And so instead of going into further examples, and I know we're running out of time here, I just want to say that with this combination of the microfluidics and the three-dimensional hydrogels, there are a lot of different kinds of mechanical perturbations and measurements that can be made to cells relevant to mechanobiology. This is just a table showing the different kind of mechanical forces that can be applied by such systems and the outputs that can be measured. Obviously, lots of flows to simulate luminal flow in ducts or vessels, but also interstitial flow, something we're quite interested in. And because PDMS is compliant, not compliant on as relevant to individual cells, but it is compliant enough that you can kind of stretch it. If you apply sufficient mechanical forces, then you can recapitulate substrate stresses and strains using the device. I have a series of slides on disadvantages, reasons you may want to think twice about using microfluidics. But since we're out of time, I can just either disseminate these slides, or you can reach out to me if you are yourself unfamiliar with microfluidics, or if you come from a lab that doesn't have much microfluidic experience and you're thinking about starting with it, then I'm happy to share some potential warnings about things to watch out for, or why you might want to consider other approaches as necessary. So with that, in the interest of time, I'll just end on this slide to ask if there are any last questions, but also encourage you to please reach out to me directly if you have any questions about what we just talked about, or any future applications. I'm happy to discuss them.